So I've been told to have to stand ridiculously close.
Oh, that's right. Well, why don't you you can hold that, can you? Oh, no, that's...
Yeah, that might work.
Yeah.
Can I use this? Yeah, okay. We'd just like to start. It's my great pleasure to introduce Gareth Howell. He's going to be talking to us about his work on behalf of Model AD and about the work that they're doing understanding the next generation of preclinical models of late-onset AD. So thank you very much to Gareth for flying over to speak to us today. We're very lucky to have Gareth today. And thanks very much. Please
okay.
Start.
Thank you. Can everybody hear me? Because I'd like not to have to hold this. Is that good? And you can hear me wherever I'm being beamed to. It's a pleasure to be here. I've never been to UCL. It's exciting to come and talk about what we're doing in Model AD. If you remember anything that what I talk about, remember that Model AD is creating new animal models. It's very open science, so we're very into sharing, and we want to get the information and the mice to people who need them as quickly as possible. So if I don't cover anything that you want to know more, then feel free to email me or any of my colleagues that I'll mention. I'm not going to give an introduction to Alzheimer's disease because I figured you probably already know and I didn't want to waste time. But what I do want to highlight is the fact that we've used animal models and particularly mouse models of Alzheimer's disease for many, many years. Karen's obviously one of the key people in the field. That hasn't translated to the clinic. And really, even despite understanding an awful lot of the biology of Alzheimer's disease through these mouse models, we really still haven't got that connection between the key hallmarks, which would be amyloid and tau and neurodegeneration. And there's many reasons why this may be. It may be that we've been focusing on early onset and late onset. The more common form is actually quite different. But it also might be the techniques, the methods, the mice that we've been generating. So within Model AD, we're looking at kind of changing it around. We're not the only ones, but now thinking about how do we kind of more closely follow the genetics of human Alzheimer's disease in order to introduce, hopefully, those pathologies in the mouse in a way that we can then study them and identify new treatments. Model AD came out of an NIA summit, so the National Institutes of Health at the National Institute on Aging in 2015, and I was lucky enough to be on the panel that was proposing better ways and new approaches to make mouse models. In general, mouse models were being created in individual labs, but then no kind of central resource to look at all the data that's being generated in human AD and then look at the ways in which we might make those mouse models. Kind of also culminated in really two amazing changes. As students, you may know that you're living in this kind of era where human genome has changed the way in which we're now thinking about complex human diseases, and also the way in which we can engineer the mouse has changed significantly, even in the last 5 years. So we can now understand the complexity of human diseases much more readily and then engineer the mouse in ways that can ask many more questions for much, much less money and much more quickly. And so those two things come together to allow us to use the approach that we're using in Model AD. Model AD sits as part of an open science series of consortia within the NIH, really looking at better understanding human Alzheimer's, so the Accelerated Medicines Partnerships for Alzheimer's disease. And this has been going, it's in its second iteration now, so it's been going for about six to 7 years. And so this is really going beyond what is defining Alzheimer's disease at the level of amyloid and tau and cognitive decline, but really what are the molecular changes that may be more relevant early. So looking at what might be druggable targets prior to the onset of any cognitive changes. So it's looking at biomarkers in the blood, it's looking at neuroimaging. There's a whole variety of different ways in which we're now, in some ways, redefining Alzheimer's disease, and this is very helpful for Model AD. It's recently created these Alzheimer's disease new medicine centers, which is now taking the work coming out of AMP-AD and identifying genuine potential, druggable targets. And so some of the people that are in Model AD are also in these new centers. And then finally, Model AD, which is really charged with generating the mouse models in which to test those druggable targets. There's more consortia to the right of this, which is more towards clinical trials. So they do have a whole long series of hours. But the one that's most relevant to today in Model AD, AMP-AD, we use a lot of AMP-AD to match our mouse models back to the human condition. And then also these new medicine centers, which are going to leverage Model AD models in order to test their various hypotheses. But that said, even if you're not part of these consortia, we're very open to hearing about new targets that might be relevant and even new models that we might need to make in order to test those those hypotheses. So Model AD has a series of goals, and I won't read them all, but really it's to take combinations of late-onset Alzheimer's disease variants, put them into mice and ask how similar does that mouse now look to the human condition. So it's going away from trying to drive the pathology that we know about in the mouse by targeting the gene itself. So traditionally we've thought about driving amyloid by mutating APP, or driving tau pathology by mutating tau. In general, in the human condition, most Alzheimer cases, that's not what happens. And so what we're looking at is perturbing variants that are relevant to the human condition in the mouse to see if we can drive those pathologies. If that's unsuccessful, then we're not averse to adding in those mutant forms, because maybe the mouse is significantly different to the human, that we may still need to drive those pathologies. But at the baseline is, what does it take to drive amyloid pathology and tau pathology in a mouse? And I'll get to the specifics about what we're doing. It's really two grants. So it's a resource grant to Indiana University, Jackson Labs, Sage Bionetworks, and University of Pittsburgh. So that's one grant, that's one consortium. And then also University of California, Irvine, led by Frank LaFurla and Andrea Tenner. But we're working together in some aspects, and as you can see here, it's divided into three cores, which is a bioinformatics and data management core, or the BDMC, and that's really taking all of that human data and trying to predict what would the combination of variants be, and also then taking the data back out that we've generated in the mouse and matching it back to human. The disease modeling project, which I lead, which is really making the mice, phenotyping the mice, making all that data available. And then a preclinical testing core led by Paul Torito at Indiana and Stacy Rizzo now at Pittsburgh, who are then taking those mice and asking, what are they a good preclinical model for? What might be the drugs that we would do it? Really establishing a stringent and rigorous pipeline for doing preclinical studies. Traditionally in the mouse, again, preclinical studies are performed maybe by a postdoc or a PhD student, doesn't necessarily have the bandwidth to do all the different things that should be done in a preclinical trial. And so that's one of the things that is being developed there. One of the very first things we did, we've been funded for about 4 years now, and one of the first things to do was we used the 5x FAD, which is 5 times familial mutations, deposits amyloid very early and progresses to about 12 months. A little bit of neurodegeneration, but not very many cognitive changes. But really just to establish the pipelines for matching our mouse data to the human data. So again, we're moving a little bit away from some of the traditional behavioral assays, although we are doing behavioral assays, and more thinking about how is Alzheimer's being defined, particularly at early stages. And so we do a lot of PET MRI in the mice. So we've got AV45, we also have tau. We're also looking at ways of developing inflammation markers in the brain. We're also doing biomarkers. So for instance, looking at A beta levels in the blood and the CSF. And then obviously we're still doing neuropathology, which is kind of the low-hanging fruit. And so it's the bit that takes the longest. We're doing a lot of omics, so a lot of RNA sequencing, metabolomics, proteomics, because a lot of the AMP-AD data is also that. And so really thinking about how we're defining Alzheimer's disease more at the molecular and protein level than just simply is or amyloid present or not. So in this talk, I've divided it into 4. I have no slides for number 4, but I wanted to tell you that that's a bit of what I do. So I'm very interested in what goes beyond the genetics. If you're interested, you can email me. There's a couple of papers we published last year on this. The third one I'm hoping to get to, but I don't have to. This is single-cell analysis of microglia from genetically diverse mouse strains. So a lot of what I'm going to talk to you in the first 2 parts are all on the C57 Black 6J genetic background, because we wanted to standardize a genetic background to ask how the variants work. But there's lots of good evidence now to suggest that mouse strains will vary the effect of the particular variant. And so we're really interested in that. And if it doesn't get to 1:45 by the time I talk, I'll show you 7 slides in 30 seconds. But really what I want to focus on is the first part is building these new strains, what we're calling platform strains, and then trying to identify variants that are relevant to human AD. So when you think about human AD, the first thing that comes to mind is age. And traditionally what's been missing from a lot of the Alzheimer's strains is an aging component. Amyloid deposits very early. So we want age to be a central component to our strains. And so we want pathology to develop across age. And then secondly, one of the greatest genetic risk factors for Alzheimer's disease and longevity, and also some of the other dementias, is ApoE4. And as I was talking with Karen earlier this morning, we don't know everything we need to know about ApoE4. It's a critical gene, and so we've set that as one of our baseline genes. The idea is we're going to layer on genetic risk factors that will tickle the right pathways in order to end up with amyloid and tau pathology and ultimately neurodegeneration. So we started with C57 Black 6 and we engineered ApoE4 into the human locus. So mouse ApoE is too different to human ApoE, and this has been done many times before with somewhat restricted licensing and use agreements and stuff. So within Model AD, we want to be able to be as free as we can to distribute these mice. So we essentially followed a previous strategy and humanized the 3 exons of ApoE, mouse ApoE, to make human ApoE4. For ApoE biologists, we've also then gone and created the allelic series of ApoE, with ApoE4 being the risk, ApoE3 being neutral, and ApoE2 being protective. And this was generated through CRISPR, so the humanizing was done by ApoE4, and then we CRISPRed to make ApoE3, and then we CRISPRed ApoE3 to make ApoE2. For those of you who don't know anything about ApoE and don't care, it's because of the amino acids that drive the different alleles. We also humanized TREM2, a recent variant, relatively recent variant now, R47H, which is a rare variant but quite a strong driver. So within the Alzheimer's field, we don't have many strong risk factors. So we've taken TREM2 R47H. It's an inflammatory gene, so it's an immune-relevant gene. And with microglia potentially being central to Alzheimer's disease, we wanted to tickle that pathway as well. As thanks to Frances initially and now work that we've verified, obviously her data, we're also correcting an aberrant splice site which gets created in this TREM2. I'm not going to go into too much detail because I could be here way past 2 o'clock talking about TREM2. Bottom line is R47H reduces the transcript levels by 50% in the mutant form due to a nonsense-mediated decay, because every now and then it splices into an aberrant splice site. By removing that splice site, making it more human-like, we can restore levels of TREM2. So if you're really interested in TREM2, and I'm talking to Frances later, I know she is, talk to me more. But basically, we're making a slightly better form of TREM2, R47H, which has now been born. So essentially what we've created, and we're calling this the first platform strain, is mice that carry ApoE4 and TREM2 R47H. And so this became our sensitizer strain, if you like. A lot of what I'm going to show you is kind of timelines and how we're going to do it and where the data might be available. As I said, we're doing pathology, biochemistry, PET-MR, network analysis like RNA sequencing, initially 5x FAD at multiple ages. But really the first big thing we did was this kind of out to 24 months, ApoE4, TREM2, and all the controlling genotypes, ApoE4 alone and TREM2 R47H. We're pretty much done. We're writing up papers. I'm going to show you 2 very quick bits of data on this. The first one is, I mentioned aging is central, so we track aging, and Stacey Rizzo developed a frailty score, which is a little bit like what you might think of as a frailty score in humans, but in mice it's obviously going to be different. They're furry, they're small, they have 4 legs. And so we do things like track the coat color, we track some of their early responses, but really it's a 1-minute frailty assay that's very easy to do, and if anybody's interested, we can get you the detail. There's about 20 or 30 things that you just look at in the mouse, and you can develop this frailty score. It increases with age, and so it's age dependent, which is good. And you can see between males and females with the different genotypes, they age the same way. So obviously we don't want to, if we're affecting aging, we need to know that, and this is a quick way of looking at whether we've impacted the aging process. We've also done a cognitive assay. As I said, cognition and more sensitive ways of looking at different types of cognition isn't central to Model AD, but we are very interested. When we see a strain, we will be able to go in more detail. Percent alternation in a Y maze, three arms of a Y maze. Mice ordinarily explore in a particular order. If they explore differently, we can capture that in this assay. It's hippocampal dependent. And so, as you can see, no cognitive deficits in ApoE4, TREM2 mice, or their controlled genotypes out to 24 months of age, which in some ways is good. It would have been nice if ApoE TREM2 alone was the strain that we needed. We could have moved that to preclinical testing. But of course, Alzheimer's is more complex than that. So now we need to think about what can we layer on. There is an interesting phenotype at 12 months that emerged from MRI. And this is a slightly complicated slide. In the interest of time, I'm going to cut to the chase, which is basically you can measure perfusion and metabolism of glucose using copper PTSM and FDG respectively. And what you see is there's an age-dependent perfusion deficit in ApoE4 mice compared to B6, suggesting something different with the blood vessels in the brain. There's also an age-dependent change in their uptake of glucose, but they don't necessarily match. So what we think of as an uncoupling of perfusion and metabolism in ApoE4 mice. And this might indicate a breakdown of neurovascular unit, which we're still following up. So in this slide here, you can see that there are ApoE4 mice. It's actually, if you think of the double, I don't have the data, TREM2 can correct this uncoupling, but it's also region-specific. So here is where were the changes particularly observed as you go through the brain. So these are the p-values. And so again, lots more information under this, but basically there's an uncoupling of perfusion and metabolism in ApoE4 mice. This may be due or correlate with a known phenotype, which is vascular decline in ApoE4 mice. This is now comparing ApoE3 to ApoE4 mice at 12 months. So my PhD student, Kate Foley, just labeled blood vessels, both basement membrane with Col4 and then fibrin. So fibrin ordinarily sequestered in the blood, but when the blood-brain barrier breaks down, you begin to see fibrin in the parenchyma. And so levels of fibrin is indicative of blood-brain barrier health, and you can see that in ApoE4 mice compared to 3, you see an elevation of fibrin levels. We're doing this by more sensitive measures looking regionally, but essentially there are phenotypes that are relevant to AD-related dementias and vascular dementia in mice carrying ApoE4. She's also now got a study where she's exercising them, and we're very interested in this idea that exercise is beneficial, but your genotype impacts how beneficial exercise is, and we know nothing about that. As a society, we should exercise more, but as an individual, does it make any difference? And so that's something that we're looking at. So I mentioned that we've made this double. The next thing that we did, and we should have really done this first, but it was the order in which we did it. The mouse A-beta sequence isn't amyloidogenic as human, and so we've humanized 3 amino acids in the mouse A-beta sequence, the R, the Y, and the H, in order to make it more amyloidogenic. No mutation here, no Swedish mutation or anything driving amyloid, but just making it more human-like. And so this becomes our triple, which is ApoE4, TREM2, R47H, humanized A-beta. Frank Lafeurle's lab have actually generated a humanized A-beta. This is a little bit like the SIDO model that some of you have been using, but it has no mutations in it. If you simply age these out to about 24 months, you begin to see diffuse plaques. They're not neuritic, so they don't stain with things like LAMP2, but they do have some evidence by silver staining here of diffuse plaques. So that's the beginnings of what we might be interested in. No other mutations here, so no other risk factors, genetic or environmental. So the thinking is in the triple, maybe with ApoE4, with some of the phenotypes, you may drive more of the A-beta-like phenotype just by simply combining humanized A-beta with ApoE. And I'm sorry for not saying beta, but I've been in the UK. I've been in the US for 15 years now. People think I'm Australian because I have an English accent with an American twang, and I get called Australian. That's either good or bad, depending on who you are. So this is the timeline. We're just starting these cohorts. Obviously, these things take 2 years. We're going to hopefully get some data after about 12 months, but the 2-year time point is going to take us 2 years. So, another thing to realize about studying Alzheimer's disease in some of these models is they're going to be longer. The studies are going to take up to 2 years. So we have to be prepared for that, both us as maybe mentors but also funding agencies in terms of how long these are going to take and how expensive. Everything's males and females. It's an N of 10 to 12 per genotype because sex is a very big confound in Alzheimer's disease, and so we need to take everything into account. We've just started putting tau in, so the 4th strain and 5th strain will be tau. Now, mice differ in tau as well. They have different ratios of isoforms, as Karen knows very well. And so we've humanized the tau region. UCI is doing it. A couple of other centers are doing it. We don't need to make the allele ourselves. If people are willing to collaborate, we can bring those in. And we're talking with Michael Kube at University of Minnesota to bring in the tau strain. He's making it available through JAX. Again, no mutations in tau yet, but we are looking at adding mutations to tau. Okay, so that's what we think of as platform strains. So those are really interesting and maybe sufficient. The final one maybe might be sufficient to drive amyloid and tau. It may not be. But of course, Alzheimer's disease is likely likely happening due to a combination of risk factors that are more subtle than those more obvious things. So if you were going to make platform strains, those are probably the ones that anybody could have thought about making. But of course, the big question is, what did the GWAS teach us? What did the genome-wide association studies teach us that we need to also incorporate into modeling? And this is not just driving the phenotype, but also understanding the biology. So why ABCA7, complement receptor 1, why did these genes strongly associate, even at a low level, at a small increase in risk? Is that relevant? Is the biology relevant? More importantly, now we've got these platform strains, is their effect greater? And so we can see whether they do or don't drive these phenotypes. There's a huge amount of data now we can leverage. We've learned an awful lot from IGAP and the versions of IGAP, Julie Williams and many, many others. But really, both in the States and around the world, there are case control studies. There's from the genetics, there's omics, so we can understand what a human brain might look like, both at the imaging level and the transcriptome, proteome, as well as cognition. And some of these are driven by NIH-funded consortium like Alzheimer's Disease Sequencing Project, the ADSP. I've talked about AMPAD, the gene consortiums, including IGAP, ADNI. And so there's loads of data that we can now take to better understand those risk factors. Where we've struggled is you've got a loci, or you've got loci from GWAS, but what should we target in the mouse? We want to be precise in the mouse. Gene knockouts may help us understand the biology of the gene, but may not help us understand the association of a variant to the phenotype they're interested in. So Greg Carter in the BDMC has worked really hard on identifying variants of those genes that might be relevant. And if you see a gene you're interested in, let me know. But really the take-home from this is we're finding variants in genes that sit in multiple biological processes relevant to Alzheimer's disease, particularly the immune pathway, lipid homeostasis, vascular health, mitochondria, and metabolism. And we've been able to prioritize variants from multiple studies that has an effect on the function of the gene, that has conservation between humans and mice, and also differential expression in AD. I said prioritize because we don't know for sure that these are the causative mutations, but we can be, Greg sometimes calls himself a sloppy geneticist. You just need predictions in human to then go test in the mouse. Because we're at JAX, we're able to make more mice maybe than others. It's cheaper for us to do these types of experiments. And so we can now ask, does that variant change Alzheimer's in a way that we think is relevant? We're hoping that this is predictive, at least improving the construct validity. So when you think about a mouse model, it's construct validity. Did you manipulate the gene in a way that is relevant to human? Face validity, does it look like the human? Predictive validity, does it reflect clinical changes? And basically we have this filtering now. So we take a platform strain, whichever one was available, we CRISPR on top of that platform strain these putative causative variants. We have a filter through a nanostring. So this is a 770 probes that we can ask, how does it look based on human? And I'm rushing because I've got about 2 minutes left. 4, 8, and 12 months. These are the mice we've made. So if you're interested in particular variants, ABCA7, some of the well-known ones from IGAP, as well as novel ones. The list is growing. We're making about 24 of these, making about 8 a year. We've currently got about 15 or 18 in the pipeline. We've done 5 of them. MTHFR is a gene that I'm personally interested in. It's really quite a common variant in the normal population. It's not enriched in Alzheimer's disease, but it's very relevant to vascular changes, and with the improvement in the understanding of Alzheimer's disease, vascular contributions are important, as well as vascular dementia and mixed dementia. So my lab is personally very interested in MTHFR. When we make one of these mice, thanks in some ways to Francis, we now do a strain validation. Does it change expression levels? That comes very early now, both at RNA levels and transcript levels. And for genes that we're actually interested in, personally, like for the biology, we can show that this on the end is an enzyme activity assay done by Rima Rosen and my postdoc, Elena Reagan, which shows that in humans, MTHFR carrying the variant has a 50% reduction in enzyme activity. The same thing happens in the mouse. So we've got additional data for this. In a nutshell, and I'll finish with these last couple of slides, what we want to do is ask, does that variant tip a platform strain closer to Alzheimer's disease than the platform strain alone did? Okay, that's the nutshell. We do that by molecular assays, which is an RNA sequencing-based assay taken out of the human data. So AMPAD spent a lot of time developing modules representative of human AD, and it's on this slide. Particularly, the one easiest to look at is this one on the right. So these are modules. It's a form of WGCNA, weighted gene co-expression network analysis, but it's actually a little bit more complex than that. Basically, all these modules down the far side are different colors representing different genes. The genes overlap, so the modules group. So you get these groups of modules that you can then enrich. So within these modules of post-mortem AD patients are astrocytes, microglia, neurons. And basically what we're going to do is take these modules, find the equivalent mouse genes, and interrogate the mouse brain using a nanostring assay that we've developed. And so this is what it looks like. Very complicated slide, but the modules are now across the top. And when we interrogate mice at different ages, 2 to 5, 7 to 9, 12 to 14, you can see that things change. You want to see blue. So the simplest prediction is the best mouse model of Alzheimer's will have blue all the way across these modules. As you can see, some of them start red. I mentioned about a deficiency in TREM2, so they start red. Red is an anti-correlation. So the genes are differentially expressed, but in the opposite direction to what happens in human Alzheimer's disease. Blue is a positive correlation. So the biggest positive correlation we have right now in the platform strains, plus the 5X FAD, is the inflammation pathways in 5X FAD driven by amyloid. But what's intriguing on the 5X FAD is none of the other pathways show any correlation, which might be one of the limitations of 5X FAD. It doesn't drive those downstream pathologies that you want, such as tau pathology and neurodegeneration. So then what we can do is we can now return to baseline, so ApoE-TREM. So now these next data is compared to ApoE-TREM, and we can take ApoE-TREM versus ApoE-TREM carrying ABCA7 or MTHFR, and you can see that now we're getting some blue. So both ABCA7 and MTHFR change some of the pathways, some of the genes in the same direction as an Alzheimer brain. Clearly not all blue, so we've got more work to do. But this is at 12 months, so you can see it's age dependent as well. So what we're now doing is adding these as our deep phenotyping. So it's gone from a primary screen, we're going to do about 24 of those, into primary screen with ABCA7. And we're putting this on the background of the triple ApoE, TREM2, humanized A beta, because obviously if we're going to wait for 2 years, we want to see if we get amyloid beta. So this is now the 3 strains that we've prioritized, including a risk variant in PLCG2, which Greg identified. These mice are just now getting organized. Invite me back in 2.5 years, and I'll tell you what the 24-month time point looked like. So this slide, in a nutshell, summarizes Model AD. We've looked at some of the original strains, and we've standardized some of the data. That's available, papers being written. We're generating platform strains, which we think will be important, and these may well be sufficient for preclinical studies. What we're really doing now is taking those platform strains, combining them with GWAS hits in a screening approach and prioritizing some of those, phenotyping using human relevant assays. All the data is available. Model AD has a website, modelad.org. All the data is going to synapse.org. All mouse strains are available through JAX. ALZform, for those of you that track mouse strains through ALZform, are also putting our strains onto their pages. I'm going to show you one slide for Soyon's benefit. Mice are different. The B6J mouse is one mouse. It's like I'm different, you're different. And so we've done Alzheimer's in different mouse strains. So we took particularly wild-derived mouse, which are very distinct. They capture almost as much genetic diversity as you see in the human population. And if you use these mice, the phenotypes are really very different. Microglia here respond differently to plaques. You can see WSB, there's almost no response to plaques versus the B6. By RNA sequencing, they respond differently. And again, you don't have to understand this, just listen to one mouse strain may not be sufficient. B6J may not be the strain that we should be using ultimately. So within Model AD, we're putting some of the variants that we see into these different mouse strains. But the one I thought was very interesting to me, so we did associated mechanical sorting, essentially, of microglia from these 4 different mouse strains carrying amyloid. And if you then do the analysis of the microglia, and strain is the greatest driver of variation in microglia with or without amyloid. So you can do tricks with the bioinformatics to then merge them together to then identify functional or potentially functional clusters, or at least the molecular clusters. So that now is from the strain differences, what are the clusters, and you merge them together. And if you then look at the types of microglia, it was originally myeloid cells, and then we prioritized the microglia. But the slide that is really intriguing is that you can see here there are different levels of clusters depending on the strain and also depending on the genotype. And the one that we're particularly interested in is these disease-associated microglia. So as you add amyloid to a B6 mouse, you get a lot of these disease-associated microglia, CAST, PWK. WSB, you really don't see that increase in disease-associated microglia, which reflects what you see with amyloid, which also correlates with neurodegeneration in WSB mice. So you don't see nerve cell loss in B6, but you do see it in the cortex and hippocampus of female-specific, female WSBs. And so we think now when we're thinking about the biology of microglia and ultimately Alzheimer's disease, we don't only need to think about different genetic variants relevant to human, but how can mouse genetics help us better understand the function of the particular cells, in my case, particularly microglia. There's a lot of people that are doing this. There's a lot of work, Indiana University, Sage, JAX, etc. And these are my own people that I highlighted. Kate on ApoE, Stanley, who's done the single cell, Elaine is working at MTHFR, and Kristen has essentially established wild-derived strains and the analysis at JAX. And that's where I work, on a small island in Maine. Sorry it was rushed, but thank you very much.
So I just want to thank Gareth for that absolutely fantastic talk, and also for speeding through it. We really appreciate that. Has anyone got a very quick question? Just yeah.
Your frailty score, is it published anywhere, or how do we get it?
If you email me, I can email you. I think it is published somewhere. But yeah, Stacey Rizzo is the one that developed it, and we can definitely provide that information for you. Yep.
Right on your variant groupings, you had your immune grouping, your lipid grouping, you had synaptic signaling. Now that's not sort of thought to promote amyloid potentially, or I mean, how do you think that factors into the disease?
Yeah,
there's a lot of genes there.
Yeah, we don't know yet. We're waiting to see what the so the way we generated the groupings was we took the genes and we enriched the genes that we were working on rather than thought about the biology and put the genes into that diagram. So there's genes that are relevant to synaptic signaling that were identified by the unbiased analysis. And so we're waiting to see what those mice look like. One of the things we're really interested in is if we've made a variant in a gene that you're really interested in, the value added from having somebody interested in a particular gene, even though we identified it in an unbiased way, is really valuable. You can see that I'm interested in MTHFR, but we've had Scott Small take SORL1 mice, for instance. And so if there's a gene that you saw, we'd love to provide some tissue. We're banking quite a bit of tissue for these individual mice. We're basically doing that primary screen, but not really looking beyond that. So if you're interested in a gene, they're all on the modelad.org website. Have a look. We'd love to add a particular collaborator to a particular mouse model.
Just very quick. Just very, very quick.
Yeah, I just thought it was really important that you highlighted vascular defects, because I think it's one of the commonly people don't really think about looking at it. And as you're, screening through this multiple, late onset, newly made GWASs, do you have any plans to look carefully at, vascular function in these hotspots, especially CR1, which is not expressed in the brain, and in others?
Yep. So I'm adding a vascular bias to Model AD, which is my own interest. So we have a staining combination that is capturing blood vessels and leakage. We're also FDG is done routinely, and copper PTSM is done if we think it's relevant. By MRI, so we do have those. I have humanized CL, so in the lab, in the Howell lab, I humanized CL1, and that's a really interesting mouse as well. We think it recapitulates some of the expression patterns of human CL1 that's not there in the mouse. So in that sense, submitted a grant to the NIH, and hopefully they'll think it's really cool to study that mouse in more detail.
Okay, I'm just going to have to conclude it there in the interest of time, unfortunately. I'm sure we can have more discussion, but I'd just like to thank Gareth again for coming to us to speak today. Much appreciated.
Thank you.
And can we all leave the lecture?